Back everyone to the first session of this second day of the ABL Annual Online Conference 2023. Today we'll start with a plenary by Dr. Renée Borges on behavioral networks in microcosms, figures, plant ants, nematodes, and fungi. So a whole lot of different model organisms. Renée received her PhD in biology from the University of Miami in Florida in 1989, where she studied resource heterogeneity and foraging ecology of the Malabar giant squirrel Atufa indica. During her career, Renée has held various positions, including visiting faculty at the Wildlife Institute of India and deputy director of the Bombay Natural History Society. Since 1997, she has been developing her research at the Center for Ecological Sciences at the Indian Institute of Science in Bangalore. She started there as an assistant professor and became a professor in 2011. Her lab is broadly interested in the evolutionary ecology of species interactions, and in particular, the sensory biology of these interactions at different levels, from chemical modalities to visual cues. Thank you, Renée, for being with us today, and you can start your presentation if you're ready.
Thank you so much. I hope I'm audible, and I'm very grateful to Animal Behavior Live, to extend this invitation to me to share the work that we've been doing over the years on microcosms. Now, there are many types of microcosms. Here is a picture of the institute where I work, the Indian Institute of Science, which is an intellectual microcosm in the heart of Bangalore city. There are other simulations of natural ecosystems that humans have tried and endeavored to create. And this is the famous Biosphere 2 experimental setup that ran for many years, trying to understand processes within larger ecosystems. Sometimes larger ecosystems are difficult to really study in their entirety. And that's why very often scientists try to work with mesocosms. And here you have examples, for example, the ecotron in Montpellier, or underground earthtrons in Japan, or oceanic planktotrons in Germany. So here you have different scales and different types of representations of the natural world. You can also have microcosms, which are artificially created. And here, of course, you've got the classic petri dish or the flask microcosms that have been used in microbial ecology quite effectively, also to measure long-term evolution. There are many natural microcosms. For example, you have the created by the symbiosis between the squid and Vibrio bacteria that create a light organ. You have the iconic pitcher plant microcosms that have so many organisms of different shapes, sizes, belonging to different phyla. And these have very, very unique adaptations. Some of these organisms are not found anywhere else other than in the pitcher plant microcosms. You also have microcosms that occur in things like bromeliads. And here you have unique species of mosquitoes, for example, that do not occur anywhere else. So many ecologists study macrocosms, but if you zoom in from a macrocosm and then come into microcosms, you will see many replicates, so many new experimental setups similar to the petri dishes and the flasks that are the artificial microcosms that we have talked about before. So this is one of the microcosms I'm going to talk to you about today. This is a closed inflorescence of a fig species, And what you see here are fig wasps, the pollinating fig wasps that have traveled many, many kilometers sometimes, and they are trying and struggling to enter this microcosm. This microcosm is technically called a syconium, and this is a nursery for both seeds that will develop because pollinators, these are the pollinating fig wasps, they will carry pollen and therefore seeds will develop over here, and wasps will also oviposit, and so this becomes a nursery for both the developing seeds and the wasps. Just like any structure has to be defended, here we have a classical example of fort walls and buttresses and other armaments. In a microcosm, you also have different occupations and different people doing different tasks. You also, as I said, have weaponry in order to defend the microcosm. And sometimes you have, doors that shut that don't allow entry into a microcosm only within certain hours. Similarly, in the microcosms that I'm going to be talking about, particularly one of them, these are the microcosms of Ficus racemosa. You have many, many, many replicates of this. Here you have a fig wasp that has come in through the entrance gate. This is a very constraining entrance gate because the wasps struggle through the bracts, they lose their antennae, they lose their wings in this process, which really makes entry into this microcosm quite a difficult task. Here are some of the people who have helped me over the years studying these microcosms, and I'm really very grateful to them. So as I've said, this microcosm gate provides a one-way entry only. So here you have a fig wasp, a pollinating fig wasp that is burrowing its way through the scales of this entry. And because it's a one-way entry, as I said, they lose their antennae, they lose their wings in this process. There is a very high premium, therefore, on correct microcosm identification. These wasps cannot enter and realize that they've come into the wrong microcosm. They can't, for example, say, oops, I made a mistake, because there's no going back. Therefore, we know from work in my lab and other labs that this attraction to microcosms is chemically mediated. Here I've just color-coded all the volatile organic compounds in a different color that are produced along the different stages of development of the microcosm. So here you have the pollination stage. I'll tell you a little bit more about the wasp dispersal stage. You have the intermediate stage where the seeds and the wasps are developing, and you have the fruit dispersal stage. So totally we have, in this model system, we have about 55 volatile compounds being produced. And as you can see, there are many more compounds produced in the earlier stages compared to the later stages. Now, Ficus racemosa is just one of the 800 or more fig species that are present in the world. And we know that most of fig species have a unique pollinator species. Therefore, this is an extreme specialization. Now, the problem also is that many of these fig species occur sympatrically. That means they all occur together in the same habitat. Now, if you've got fig species that are producing volatiles, and many of these volatiles, some of them are quite similar. All that differs is probably a ratio or the nature of the blend of the compounds. And therefore, one might view this identification of the particular blend that is calling or is, produced by your particular species. So here you have fig wasp species 1, fig species 1, fig wasp species 2, fig species 2. This problem is similar to the cocktail party effect that is very popular in the auditory system, where people, talk about a lot of background noise, and species have to pull out from the background noise the particular signals of their particular channel of communication. So here, obviously, this is an important problem to be solved. And when we have looked at sympatric species and mapped their volatile organic compounds that are produced in the stage when the pollinators are coming into the microcosm, we find that it maps very well and separates out in multidimensional space. Of course, we've also done experiments where we give pollinator species scents of different fig species to choose from, and we find that they make the right choice. So here is another way that pollinators are, very sure, or they have to be sure, that they are entering the right fig at the right time. Now, we've also done work on olfactory receptor genes, and if one looks at Apis mellifera, for example, Apis mellifera has 162 olfactory receptor genes. But if you look at the pollinators of figs, we have 2 species for which the olfactory receptor genes have been annotated and found out. We have about 63 in Ficus hispida and just 56 in Ficus racemosa, which means that because of the specialization of this system, most olfactory receptor genes are either pseudogenized or they are absent. Another interesting finding that we've discovered is that there is ectopic expression of these olfactory receptors. And as a matter of fact, we find, yes, there is, of course, expression of these receptors in the antennae, but we also get expression of olfactory receptor genes in a key organ like the ovipositor, and I'll talk a little bit more about that later. So for now, remember that olfactory receptor genes are also expressed in the antenna of these fig wasps. Now, I said that there was a legitimate way to enter these microcosms. Is there any other way to gain entry? And yes, there are parasites of the mutualism between the fig and fig wasp. And these parasites don't enter, the whole body of the wasp doesn't enter. It's only the pollinating wasp that comes in. But these parasites pierce into the microcosm using these very long ovipositors, and that's how they can also be gallers or they can be parasitoids, laying their eggs on the developing larvae of the other developing gallers in the microcosm. So here, just to see how it looks like, you have seeds that are developing along the walls of the microcosm, and in uniovulate flowers, which have become galls because of the action of the oviposition of the pollinators, you get the developing gall larvae also within. So now, as I've said, you've got pollinators coming in through the ostiole, the syconium develops, it becomes larger. You have gallers that are exploiting the system early on. They oviposit even before the pollinators come in, and you have even late arrivals. So parasitoids that come in much later. Interestingly, all of them have to leave the microcosm at the same time, because the males of the fig wasps that are wingless, these are the ones that cut an opening for the female pollinators, and the female pollinators leave the microcosm carrying pollen, and simultaneously all the other parasites take this opportunity to leave the microcosm, and this is a stage that is called wasp dispersal. So in our community, we've got the pollinator, that is a galler, we've got three other gallers, and we've got three other parasitoids. So here is what the community members look like. The female and of the 7 species, and the males of all the 7 species. You can see that there is an amazing sexual dimorphism. Males are wingless, and they are born and they die within the microcosm in which they are. Now, we've also looked at what we call the oviposition windows, when the females can colonize the microcosm, and we can see that as the microcosm is growing, you also have the ovipositors that correspondingly grow in size. Now, every microcosm is prone to parasitism. I've told you about the parasitoids and the parasitic gallers. We also have other players in the game, and these are ants, and these are eavesdropping on the scent of the microcosm. So here you've got 2 species of ants, and you can see the pollinators are trying to rush into the microcosm, and the ants are trying to pick them up as prey. Now, we've done experiments with them and have shown that the ants are sensitive to the volatile organic compounds that are produced by the fig syconia at the time when they are releasing the scent to attract the pollinators in. And in olfactometer experiments, we can clearly see that this is the B stage of the figs, which is the pollination receptive stage. And this is when ants like Echophilus smaragdina and Technomyrmex albipes, this is when these ants are sensitive and they respond to the scents of these figs. Now, we also found, much to our surprise, that this response to fig-associated odors is a learned response. So when we take ants that are naive ants of both the species and test them against these volatile organic compounds, we find that the ants are not able, they don't respond. So clearly, the naive predatory ants are unable to respond because they haven't learned to make the association between the arrival of the wasps and the fig odors. There is another very interesting component in this microcosm, and these are nematodes. Nematodes are highly specific to particular fig species, and the association between figs and nematodes is also very old. Now, nematodes also in this system exhibit what is called a very interesting behavior called phoresis. Phoresis is when a tiny individual uses a larger species to move between resources. So resources are declining in one area, and the phoretic species is either immobile or incapable of moving, then the phoretic species jumps onto its ride. So we have riders that are the phoretic species and the rides that serve as the vehicles. Now, if you want to be a ride, to get on and take a ride, you need to have what are called embarkation cues, and you also need disembarkation cues. So you need to know when to embark and when to disembark. Now, nematodes are ubiquitous as riders. So they use other species. And it is actually the Darwin life history stage of nematodes that is really the stage at which the nematodes seek out rides in order to jump onto them, to use them as vehicles. So here you can see nematodes in our fixed system doing what is called a classical nictation behavior and wanting to jump onto their rides. We have 3 genera of nematodes in our system. And what we have found is that nematodes hitchhike or use only the female pollinators as their rides. We've done experiments with single nematodes, giving them choices between male pollinators, female pollinators, giving them choices between just the volatiles of male and female pollinators. We gave them choices between the cuticular hydrocarbons extracted from male and female pollinators. And then we've given them also cafeteria choices. And we find that in all these experiments, nematodes are able to pick out the female pollinators and they ignore males of their own, of the pollinator species. And they also ignore females and males of all the other species that are present inside the syconium when the wasps are dispersing. So this clearly means that nematodes are queuing in and using cues from female pollinators as embarkation cues. We've also established that one reason why they can possibly use cuticular hydrocarbon signatures as embarkation cues is because all the species of wasps within the microcosm have very, very distinct cuticular hydrocarbon signatures. So here we have a cartoon or an illustration of this fantastic embarkation and disembarkation system. So as I told you, this is the stage when the wasps are escaping from the syconium. And this is when the nematodes have to embark, and they embark during the dower stage. And then the female pollinators have to find a syconium in the pollen receptive stage. So the female pollinators have to come into the ostiole, and I've shown you images of this. And this is what we call then the disembarkation stage for the nematodes. Now, what cues might nematodes use for embarking on live wasps? So here again, we've done a whole set of behavioral experiments. We've done single choice as well as 2 choice assays. And as a matter of fact, I think that probably our lab is one of the few labs maybe in the world that actually experiments with single nematodes. These are very, very difficult to perform because the nematodes are extremely tiny, as you have already seen in the videos. So when we do single choice assays, for example, and here I'm just showing you for 2 of the nematode species. This is an animal parasitic nematode and a plant parasitic nematode. We see clearly that the nematodes are attracted to the live wasps. Here, remember, these are single choice assays. Even if we give them dead wasps, they are still attracted to wasps, okay? Both the species. If we do 2 choice assays, you can see that the nematodes are more attracted to the live wasps than to the freeze-killed wasps or the naturally dead wasps, which means that there is something that is coming from the live wasp, which is telling the nematodes that, yes, this is a live vehicle and this vehicle is likely to be able to take me to my destination. So here you can see clearly in both species Teratodiplogaster and Schistoncus, they show more of a preference or an extreme preference for the live wasps. So then we were interested, how do nematodes know that the wasps are alive? They could be getting other cues from the cuticular hydrocarbons, etc., which won't differ much from a freshly dead versus a live wasp. And then we found that it is the carbon dioxide that is released by the live wasps that gives the cues. So we've done experiments where we've just presented carbon dioxide at the level that is produced by a single respiring wasp. So we've been able to measure the μL of carbon dioxide released by a single respiring wasp, and then we have given those levels in our behavioral single nematode experiments, and we have found that nematodes are able to use just the carbon dioxide. They are not attracted to clean air at the same level, and they're certainly not attracted to carbon dioxide at levels that are lower than this. Now, I've also said that yes, there is an embarkation phase and a disembarkation phase. There is a huge struggle for the nematodes to get on to these wasps when they are leaving. But you will see that there are much fewer nematodes that are present when the wasps come back. And this is because the wasps, the nematodes really, have a big toe.
Seems like there's some technical issues and we may have lost connection to Renée. We're just quickly trying to get her back online as soon as possible. So please stay tuned. Can you hear me?
Yeah, I can.
So you can share your screen and then I just announce that you're back and then you can continue.
Right. Share my screen. Yes. Yes. Can you see me now?
Yes, it's great.
Okay.
I think we transitioned back and then I just announced you're back and then you can continue. Okay. Okay. Welcome back, everyone. Sorry, we had some technical issues, but Renee is back with us and she'll continue her presentation now.
Yeah, sorry. So, because wasps nematodes cannot predict whom they are going to encounter in the syconium that they reach on their vehicle. Therefore, they have to make sure that they have companions, but not too many companions on the wasp that they are hitchhiking on. And therefore, we can see that there are one of the species definitely avoids getting on to an empty vehicle. The other species prefers to get onto empty vehicles, but we know that this species disperses in pairs. So two nematodes will jump on to their vehicle, and this is probably why we get a difference between one of the parasitic nematode species and the plant parasitic nematode species. So, as I said, if the vehicle is the only one to enter the fig microcosm, then it will be game over for the nematode. Now, I've also mentioned that there are other wasps that enter the microcosm through their ovipositor. They don't come in themselves. But they have to also know who has previously oviposited into the microcosm. And we've done studies on both the microstructure as well as the presence of salicylae on the ovipositor. And we find that the ovipositor is really very, very different from species to species. And as I have mentioned, and I mentioned earlier, that the ovipositor of a fig wasp is behaviorally sensitive to volatile organic compounds. So we have demonstrated that the ovipositor, like the antenna, is an olfactory organ. We've done measurements. We've modified an electroantennogram into what we call an electro-ovipositogram, and we've been able to show that there are sensory neurons in the ovipositor that pick up volatile organic compounds. Now, we often get large aggregations of parasites on the outside of microcosms, and we might ask, why are wasps aggregating? And we believe that this is because they want to ensure that their offspring will have conspecifics to mate with once the offsprings eclose and develop within the microcosms. So we have found that the parasites leave their chemical signatures on the surface of the microcosms, and we've been able to analyze these chemical signatures. And here you can see the GCMS profiles and the identification of these compounds that different species of wasps are leaving on the surface of microcosms. We've also been able to show that wasps are very choosy as they age. So possibly just like humans, we have a biological clock that keeps ticking. Similarly, the biological clock of wasps also keeps ticking. And once they become older, they are much more are less choosy about which microcosm they will lay their eggs into. Okay, so the time that they take to oviposit into the microcosm is much, much shorter as the wasp's age. Okay, so I've given you a glimpse of the diversity of interactions that happen within the fig microcosm. There are many, many species of figs in the world, and these are beautiful examples. Wherever you are in the world, you will have many species of figs in the tropical and subtropical regions. And I would encourage you, therefore, to explore these microcosms for the plenitude of interactions that you will experience there. Now, moving on from figs to ants and plant interactions, I've also worked, and I'll give you very briefly a few examples of some of the studies we've done with an ant plant microcosm. Here you have ants, and you also have the microcosm, which is a domatium, which is a swollen hollow internode. This is the team of people, some of the people that have helped me in this work. And of course, you might ask, why are ant-plant interactions so important? Because ants are really constitute a lot of the terrestrial biomass. Many plants offer ant shelter in the form of domatia or swollen thorns or internodes. Plants offer liquid food in the form of extrafloral nectar. Plants also offer solid food in the form of food bodies to ants. This is the species that we worked on for many years, Humboldtia brumonis. This is the domatium, and here are the extrafloral nectaries from the buds and the leaves. This is a part of India where we did our work on Humboldtia brumonis in a semi-evergreen, quite a dense forest. And this is a normal internode, and this is an internode which is hollow, which is meant for the residence of the ants. There are many, many domatia occupants. We have about 16 species that occupy these domatia. And we also have an arboreal earthworm, which is very interesting, that also muscles its way into the domatia. And in this microcosm, we have a parasitic ant that destroys flower stamens and engages in what is called castration. And we also have a mutualistic ant, which is the one that actually confers protection against herbivory. Now, these ant species vary in their recruitment and reaction to interlopers, and we've done some nice experiments with earthworms and other herbivores that we've tested these ants on. And you find that Technomyrmex albipes, compared to the parasitic ants, Technomyrmex albipes has the most number of ants that are recruited to these interlopers. And it's really the one that is able to bring in a lot of workers to the site of the action. So, commensurate with this aggressive behavior, we also have Technomyrmex albipes patrolling throughout the day and night, patrolling the leaves, patrolling the flowers, compared to the parasitic ants that are not doing much patrolling. And correspondingly, we find when we do ant exclusion experiments, where we exclude ants from contacting the leaves, we find that Technomyrmex albipes is the one that is most efficient at preventing herbivory on the leaves. Now, as I've also told you, there is a whole web of interactions. There are ants, there are earthworms, there are predators. Here you see a centipede feeding on the earthworms that are present inside the domatia. Now, because there are so many interlopers, you might want to ask the question, do domatia tenants pay the rent? I mean, who's paying the rent? Are they giving back something to the plant. And very interestingly, we were able to find, using stable isotope analysis that I'm not presenting here today, that even the parasitic ant gives about 17% of nitrogen to the domestium tissue, which is then taken up by the rest of the plant. Even the earthworm, that is really a squatter in this tenement, gives about 9% of nitrogen back to the plant. Now, there is also a fungus. So I want to end by coming to the fungus part of my presentation, because I promised that I would also talk about fungi. And we know that there is a subtenant in this interaction. There is a fungus which is cultivated by ants within the domestium. Here you can see the hyphae of the fungi. And we know that there are many interesting interactions between ants and fungi. Many of them are in this group called Ketothyreales. And indeed, when we sequence the fungus that we find in the domatia, we get other fungi, but most of them belong to the Ketothyreales. So there is certainly a very close interaction that is going on over there. Now, in the last couple of minutes that I have before I want to leave enough time for questions, I just want to introduce you to another microcosm system, and this is the termite microcosm. And here are some of the people that have worked with me on the termites. Now, termites in the old world tropics, many termites cultivate fungi. And we know that farming is a very, very interesting behavior. Humans are farmers only for about the last 10,000 years. But there are insects that have been doing farming for millions of years before us. So, of course, if you want to cultivate mushrooms, you need a greenhouse. If humans have to do it, you have to maintain moisture and temperature very carefully. This is the kind of greenhouse that we need in order to cultivate fungi like mushrooms. But here are the greenhouses of the termites. So here are the termite greenhouses on the campus where I do my work. These are the fungal greenhouses. You have the fungal chambers which are within the greenhouse. And the fungi in the sexual form produce basidiocarps or mushrooms which come outside the greenhouse. Yeah. So these are the ones. In the greenhouse, they are in the asexual stage, and then they form the fruiting bodies. So here is a fungus comb, which is the asexual stage, which is cultivated by the termites. And here you can see a zoomed in version of the mycoteats, which are the globular structures formed by the asexual form of the fungi. And this is what the termites cultivate. This is termitomyces. Now, like all microcosms, there are always parasites waiting in the wings. And the parasite here is Pseudoxylaria. So if we take away the termites and prevent them from interacting with the fungi or the fungal community, Pseudoxylaria takes over. So we were interested in asking questions: how do termites keep their fungal crop pure and prevent Pseudoxylaria from taking over? So this is the termite fungus farming system. As I told you, you have Pseudoxylaria, you have the termitomyces, which is the crop, and you have the soldiers and the major workers and the minor workers. As I've told you, termites have to maintain their greenhouses, otherwise the fungus will not grow. And we have seen that termites are capable of regulating temperatures inside the greenhouse. This is outside the nest, and this is within the nest where the temperatures are constant. Very low variation in daily temperatures. they regulate humidity in the greenhouse. So this is humidity inside, kept close to 98% of humidity, whereas outside the nest, you have a huge fluctuation of humidity. Now, any farmer can control weeds by weeding, by having abiotic conditions that are favorable only to the crops, or by application of weedicide. So we wanted to know what are the termites doing. Many antifungal compounds have been identified in particularly African fungus farming termites. And here are some examples: natalamycin, spinigerin, termicine. But all of these have been found to have very broad spectrum fungicidal action. Now, if farmers are going to put a broad spectrum fungicide, obviously they will kill their fungus crop also. So how can non-specific antifungal compounds be deployed against weeds if they are also going to harm the crops? So this is the problem that we wanted to address. And it took us a long time to come up with a successful assay system. So here we have a Petri dish assay system where we put the parasitic fungus, the mutualistic fungus, and we ask the termites, we interrogate the termites and ask them, what do you like it? What would you do with these plugs of these fungi? And the assay involves, the termites have this constitutive mechanism. They cut agar and they bury these plugs. So it's a constitutive behavior, cut and dump, cut and dump. So when we, so here is termites in the act of burying the parasitic fungus. They have cut agar and they are burying the fungus. And when we do these behavioral assays, we find that the soldier termites don't do anything. They are unresponsive. The major workers are the ones that respond majorly to the parasitic fungus and they bury the parasitic fungus. So you can see in all cases, in one, we do these assays over several hours and you get most of the parasitic fungus gets deposited by agar. Now, we wanted to know, can they tell this by smell? Do the two fungi smell differently? So we did olfactory assays here where we gave them, we didn't allow the termites to actually contact the fungi, but they had to only smell the fungi. And here again, with only the smell of the fungi, you can see that the termites can pick up the difference between the mutualistic fungus and the parasitic fungus, and they dump more agar on the parasitic fungus. Now, we also looked at the GCMS profiles, and we can see clearly that there are different compounds emitted by the mutualistic fungus and the parasitic fungus. And if we do a NMDS plot or a multidimensional plot, you can clearly see that the crop fungus and the weed fungus, the weedy scent really largely has aristoline and viridiflorol. We are yet to test these compounds on in their pure form, which is something that we will do in the future. So clearly, termites can smell the presence of the weedy fungus and take necessary action to control the weeds. So in this case, when they can smell even a few hyphae of the weedy fungus, they can therefore selectively deploy antifungal compounds, and therefore, and in this way, termites can avoid poisoning their own crops. So microcosms therefore afford fascinating insights into behavioral, ecological, and community networks. I hope I've been able to show you a few examples of how fascinating microcosms can be. Of course, in my lab, there are some indispensable people without whom the microcosm would collapse, and these Sunita and Yetiraj, who really help in all our lab work. And I'd like to acknowledge, of course, my institution, the lab Syncephalon, that I call it, and all the funding agencies, and many other labs that have helped in this journey. Thank you very much, and I'm happy to take questions. I'm sorry about the glitch, but the internet went off for some time. Thank you.
Thank you so much, Sunny. That was a fascinating talk. It really covered a lot of different things. Um, I think we already have some questions on the YouTube chat, so maybe I'll start with it. We have a few questions from David. David is a part of the organizing committee and he loves wasps, so he's been looking forward to the talk for a while now. So the first question he asked is, you mentioned that the ants that prey on the pollinating wasps learn this behavior. Do you have any idea of how they learn this?
Okay, so these ants are actually quite visual also, Echophilus maragdina and Technomyrmex, particularly Echophyla. So I think that they are, patrolling. If they are on fig trees, they are patrolling them. And I'm certain that they are also visually alerted that there are a lot of, potential prey items coming in. And then once they are visually alerted and they come to the figs, yes, and then they are also able to smell this. And then they make the association between the smell and the arrival of the figs. Because in our olfactometer test, we've just given them the scent of the figs. So they have no visual cues over there. Yeah.
So his second question is if the nematodes in the figs provide any benefit to the wasps on the figs, or they produce any benefit at all to any of the organisms involved in this microcosm.
So I think that the nematodes are entirely parasitic. I don't believe that they have any advantage, but if you remember the figures I showed you of the numbers of nematodes that are present on wasps that are leaving the fig, and the numbers of nematodes that are present on wasps that are arriving. So it's clear that there is a big difference. Only wasps that have fewer numbers of nematodes make it back to a site where they can lay their eggs, etc. So there is a strong selection pressure on wasps not to carry a lot of nematodes because they will not make it to their next destination. But we still do not know whether wasps can control the number of nematodes that get on because the nematodes are very quick, they jump on. So, and the wasps have only an adult lifespan of 24 hours. So, in that very short time, they've got to find a fig tree in the right stage in order to get in and lay their eggs, etc. So I do not believe that the nematodes have. Actually, I haven't shown the data, but we've got a lot of data to show how nematodes are actually bad for the fig. We do experiments where we artificially load a whole lot of nematodes onto our experimental wasps. We find that the nematodes reduce the flight time, the flight duration. They actually reduce oviposition behavior. They do a lot of not very sustainable things for the mutualism. So the nematodes are not good for the mutualism.
So just continuing off this question, I guess then the how fast the nematode acts is like the time duration that they take to infest and act on the wasp is quite short because, as you said, they need 24 hours. They only have 24 hours to find their way back to a place where they can oviposition. And so if the load is higher, then probably within 24 hours already they've had storm effects. Is that what you see when you mentioned that you did some?
Absolutely. So we load wasps with the nematodes, and then we do all the flight assays, we do the oviposition assays, we even do ant predation assays, because you've seen that the ants are waiting for the wasps, right? To as they are coming into the osteo to enter. uh, Wasps that are heavily laden with nematodes are more sluggish. So even if they land onto the fig, they are not quick enough to enter. And therefore,
I think we lost her again. I think the power just went because I can see the lights are off. Maybe we wait a minute to see if she can join, or since we are reaching to the end, I guess further questions can be on the Discord channel. She told us she's joined, so if you have any other questions, you can ask her on the Discord channel. But we anyway just wait for the minute to see if she can join back.